Hello everyone, I am Dr. Nilanjana Bhattacharya Nath from Swami Vivekananda Institute of Modern Science. Today's topic is protein purification and tagging. That means recombinant protein expression and how to choose the right affinity tag. As you have selected a protein for biochemical studies, and if you have cloned that particular protein into a tag containing vector, so what will happen? After successfully expressing your protein in a suitable form, you need to do is pick it out from the proteome pool of the expression system. So an affinity tag, just like an identity card, it allows you to identify and separate your protein from a particular mixture. So protein purification affinity tags, they enable your protein to bond with a particular molecules and leaving behind other, that means non-tagged proteins. Basically, protein tags are peptide sequences that are attached to proteins to facilitate easy detection and purification of expressed protein. And in addition, they can also be used to identify potential binding partners for your protein of interest. At present, there is wide variety of protein tags that can be used for our protein tagging purpose. And these include number 1, affinity tags, number 2, epitope tags, number 3, fluorescent tags. So first one is affinity tags. What are they? Affinity tags allow you to purify proteins from crude cell lysate, even if you don't have any knowledge of the biochemical properties of that particular protein. And this affinity tagged protein can be purified by using a specific affinity resin, and sometimes the detection can be done by using commercially available antibody. So examples of affinity tags, that is GST, glutathione S transferase, polyhistidine tag, calmodulin binding protein CBP tag, MBP tag, maltose binding protein. So these are all affinity tag. So first I will be discussing hexahistidine tag or 6X histidine tag. Hexahistidine tags, they are one of the most widely used affinity tags, and it utilizes the ability of histidine to coordinate with transition metal ions such as nickel, copper, and basically they are having small size, they are non-toxic and does not interfere with the immunogenicity or physiochemical properties of the protein. And 6X histidine, basically this tag comprises of 6 to 8 histidine residues. That is why they are also known as 6X histidine tag. As I have told you, they are having relatively small size and makes its integration into expression vectors extremely easy. Histidine tagged proteins, they are purified using immobilized nickel, cobalt, zinc, copper ions. And the main thing is that these tags do not form secondary structures to bind their substrate. So it will be easier for us to purify our protein of interest, even under denaturing conditions. And during purification, ensure that EDTA and other metal chelators are avoided. Next one is glutathione S transferase, GST. GST is the another affinity tag, and it works on the principle of GST's affinity for immobilized glutathione. So a GST tag can increase the solubility of your expressed protein in a prokaryotic system. Since GST has a strong binding affinity for glutathione S transferase, so what will happen? Beads coated with the compound can be added to the mixture to isolate our own protein of interest from rest of the solution. And this technique can be used to explain direct protein-protein interactions. Next, MBP, maltose binding protein. So the main aim of this tag is to increase the solubility of the fusion protein. So it may be our tag of choice when purifying membrane or lipophilic proteins. MBP based purification helped us to obtain decent expression and yield of a membrane protein that was difficult to achieve with 6X his tag. So the MBP fusion protein bound to an amylose resin which is eluted by running maltose in an elution buffer, and this method is fairly resistant to denaturants, reducing agents, detergents. But any amylase activity in the cell lysate may reduce the efficiency of the amylose resin. These tags bind to the amylose agarose and are commonly used to increase the solubility of fusion proteins. Next, CBP, calmodulin binding protein. They are small size, approximately 4 kilodalton, and makes it ideal for purifying delicate proteins under mild conditions. And the tag binds to the calmodulin resin, and the proteins can be eluted with a neutral buffer containing low concentrations of EGTA, calmodulin binding protein, that CBP tag. They can withstand low level of some unwanted components such as denaturants, detergents, reducing agents, etc. These are all affinity tags. So second one is epitope tags. So these tags are typically smaller than affinity tags and are readily recognized by antibody. And due to their relatively small size, they have extremely little or no effect on the structure of the resulting fusion protein. And these epitope tags, they are ideal for several downstream applications. For example, Western blotting, co-immunoprecipitation, immunofluorescence experiments, etc. And the few examples of epitope tags are MIC tag, HA tag, human influenza heme agglutinin tag, FLAG tag. So this HA tag is a peptide sequence derived from the surface glycoprotein, and this particular glycoprotein, it facilitates the ability of the influenza virus to infect its host, and it is recognized by numerous commercial antibodies. The ability of influenza virus to infect its host and recognized antibodies. And this particular epitope tag is widely used for tagging in expression vectors, and it is useful for the detection, isolation, purification of our protein of interest. Next, MIC tag. This is also an epitope tag, and it is a short peptide sequence derived from the c-MIC gene product, and it is also recognized by numerous commercial antibodies. Next, FLAG tag. Like MIC tag, FLAG tag is also a popular short peptide sequence which is used in recombinant DNA technology and can be used for affinity chromatography and for protein isolation. It is also recognized by numerous commercial antibodies. It can be fused with the C terminus or N terminus of the protein. It is much more hydrophilic as compared to other tags, and they do not denature or inactivate the proteins to which they are attached. Last, fluorescence tag. Fluorescent tag is basically non-toxic, and these tags can be used to detect tagged protein in both live as well as fixed cell. And green fluorescent protein GFP is one of the most widely used protein tag under this category. GFP, that green fluorescent protein, it is a protein isolated from the jellyfish Aquaria victoria, which exhibits bright green fluorescence, and that fluorescence that does not fade easily when exposed to blue or ultraviolet light. That particular fluorescent tag, they are remarkably stable and function when added to either end of a protein of interest. Other than these tags, that is affinity tag, epitope tag, fluorescent tag, another tag that is very much important, that is streptavidin biotin based tag. We simply choose a particular target protein which is fused to a short peptide, approximately 9 amino acids, and known as the strep tag. And that particular peptide binds to a streptavidin. And the protein is eluted by using biotin in the elution buffer, and this system achieves a high purification efficacy. And after completion of your purification, you can remove the tag depending upon the downstream experiments or assay. So this is the important tagging system, starting from affinity tag, epitope tag, fluorescent tag, biotin streptavidin tag, etc. Thank you.